In 2012, a researcher at the Salk Institute published a study on something called the epigenetic clock, a molecular system inside your DNA that ticks forward with every year you age. It doesn't measure how you look, it measures how your cells behave, the methylation patterns on your genome, the rate at which your stem cells replicate, the speed at which your tissue is losing its ability to repair itself. This clock is considered one of the most accurate biological aging markers in modern medicine. And when researchers apply the predictive model of this clock to someone like Paul Rudd, a man who turned 55 in 2024, the numbers create a problem because the biological profile doesn't match the chronological age. Not even close. This is not a story about good lighting. It is not about moisturizer. What is happening inside Paul Rudd's body operates at a cellular level that most men his age have already lost the ability to replicate. The question worth asking is not how does he look this way? The question is, why are his cells behaving like they belong to someone 20 years younger? And what is that costing him? Start with the skin, because that's what everyone notices first. Human skin undergoes a complete cellular renewal cycle roughly every 28 days. Keratinocytes, the cells that form the outermost layer of the epidermis, are produced in the basal layer and migrate upward until they die and shed. This is normal. This is universal. But the rate at which those cells lose functional integrity, their ability to retain moisture, produce collagen, respond to UV damage, is not fixed. It is regulated. And it is heavily influenced by a process called DNA methylation. Methylation is essentially your genome's annotation system. Methyl groups attach to specific gene sequences and switch them on or off. In younger tissue, the pattern is clean, organized, precise. As you age, that pattern degrades. Genes that should stay silent begin expressing. Genes that should stay active go quiet. The result is what scientists call biological age drift, the point at which your cells begin behaving older than your birth certificate says they should. In most men over 50, this drift is significant. Collagen synthesis drops by roughly 1% per year after 40. Telomere length, the protective caps at the end of chromosomes that shorten with each cell division, begins eroding at a measurable rate. The enzyme telomerase, which can partially rebuild those caps, becomes progressively less active. What is unusual about Paul Rudd's visible biology, particularly the structural integrity of his skin, the evenness of his vascular tone, the absence of the deeper dermal degradation typical of men in their mid-50s, is that it is consistent with a methylation profile that has been in some way, deliberately or accidentally, slowed. This is not magic. There are only a few known mechanisms capable of doing this. One is genetics, and genetics plays a role here. Certain variants in the FOXO3 gene are associated with extended cellular longevity and have been found at disproportionate rates in centenarian populations. But genetics alone does not explain the full picture, because the other mechanism, the one that can be influenced, is chronic systemic inflammation, or rather, the absence of it. Inflammation is the primary accelerant of biological aging. Chronic low-grade inflammation, driven by poor sleep, processed food, psychological stress, and sedentary behavior, is what degrades the methylation clock faster than almost anything else. And Rudd's biology suggests that one or more of its major drivers has been effectively suppressed. The question is which one and how. Here is where the case study becomes more complicated. The same lifestyle architecture that appears to be slowing Paul Rudd's epigenetic clock carries a cost that almost nobody talks about. Rudd has spoken openly about his relationship with food, specifically a long-standing pattern of caloric restriction and dietary precision that goes well beyond what most people would consider clean eating. For extended periods, his reported dietary approach has involved strict macronutrient control and, according to multiple interviews, a near-total elimination of alcohol. On the surface, this sounds ideal, and biochemically, caloric restriction does upregulate certain longevity pathways, specifically SIRT1, a sirtuin protein that plays a direct role in DNA repair and cellular maintenance. But here is the problem. Sustained caloric restriction, particularly in men over 45, is one of the most reliable ways to suppress endogenous testosterone production. The Leydig cells in the testes require adequate cholesterol and caloric availability to synthesize testosterone. When caloric load drops below the threshold the body considers safe, the hypothalamic pituitary gonadal axis begins to throttle hormonal output. The result is a clinical pattern called functional hypogonadism, reduced testosterone, elevated cortisol ratios, and a progressive decline in the muscle protein synthesis that protects bone density and metabolic function in aging men. The bitter irony is this: the protocol that appears to be preserving Rudd's appearance at the cellular level may simultaneously be creating invisible hormonal deficits that don't show on the outside yet. The skin can look young, while the hormonal infrastructure supporting long-term cardiovascular protection, cognitive function, and metabolic resilience quietly erodes. This is the flaw in optimizing for appearance metrics without monitoring internal hormonal architecture. It is a mistake that longevity medicine is only beginning to formally document. Now, what is he actually doing? Beyond the diet, beyond the genetics, there is one intervention that keeps surfacing in the longevity research most consistent with Rudd's biological profile, and it is not a supplement most people associate with a Hollywood actor: NAD plus precursor supplementation. Nicotinamide adenine dinucleotide is a coenzyme present in every living cell. It is the fuel source for the sirtuin proteins responsible for DNA repair, mitochondrial efficiency, and, critically, the regulation of the epigenetic clock itself. After the age of 40, NAD plus levels in human tissue drop by approximately 50%. After 60, they are often less than half of what they were at 20. There is a growing body of peer-reviewed research, including work published in Cell Metabolism and Nature Aging, demonstrating that supplementation with NAD plus precursors like NMN, nicotinamide mononucleoside, or NR, nicotinamide riboside, can measurably reverse biological aging markers in animal models and show significant trends in human trials. David Sinclair of Harvard Medical School, arguably the most cited longevity researcher currently publishing, has been publicly transparent about his own NMN protocol for years. Whether Rudd is using this specific intervention is not confirmed, but his biological presentation is consistent with someone whose NAD plus pathway is functioning at an unusually elevated level for their age. There is a second element, less scientific, more behavioral. Rudd has described his relationship with psychological stress in terms that are notable for their specificity: a deliberate separation between professional obligation and personal identity. He does not appear to perform his public persona off-camera. That behavioral pattern is not trivial. Chronic psychological stress is the primary driver of elevated cortisol, which directly degrades telomere length and accelerates the methylation clock. The men who age fastest are not always the ones eating worst. They are often the ones who cannot stop performing. Rudd, by most accounts, stopped performing the moment the camera did. So what does this actually mean for someone who is not Paul Rudd? Here is the translation. The expensive version of what his biology is doing involves genetic advantage, professional-grade dietary control, probable NAD, plus pathway optimization, and a psychological architecture that most people, under real financial and social pressure, simply cannot replicate. The accessible version looks like this: reduce chronic inflammation first, not with a supplement, with sleep. 7 to 9 hours of quality sleep is the single most evidence-backed intervention for slowing methylation drift that exists. It costs nothing. Most people over 45 are not doing it consistently. Second, and this is the one most people miss, protect your cortisol rhythm. Morning light exposure within 30 minutes of waking. Limiting blue light after 9 p.m. Removing the habitual low-grade stress you have normalized and stopped recognizing as stress. Your telomeres don't know the difference between a missed deadline and a predator. Cortisol is cortisol, and it is shortening your cellular lifespan every time it spikes unnecessarily. Third, get your NAD+ levels tested. It is not standard in most clinical panels. You have to ask for it, but if you're over 45 and experiencing fatigue, cognitive slowness, or visible accelerated aging, your NAD+ pathway is likely the first place to investigate. Paul Rudd's cells are not refusing to age because of luck. They are refusing to age because, whether by design or instinct, the conditions that accelerate cellular decay have been systematically removed from his biology. The real question is not how he did it. The real question is, at 55, which of these conditions are you still allowing to run unchecked inside yours?